today's botany class on photosynthesis. Before going to today's class, let us first have a look at what we studied in the previous class. So, in the previous class, we had discussed about the dark reactions of photosynthesis or the light independent reactions, or also known as the stroma reactions. So, in these stroma reactions, there is reduction of carbon dioxide into hexose sugar or commonly glucose. So, there were different types of paths of dark reaction for the fixation of carbon dioxide. The first type that we saw last class was C3 type or the C3 cycle. So, the C3 cycle, commonly known as the Calvin cycle. So, Calvin cycle, the initial enzyme that accepted carbon dioxide is the enzyme Rubisco or RUBP carboxylase oxygenase. So, in the dark reactions of the C3 cycle, 6 molecules of carbon dioxide, they were converted into a molecule of glucose, and we regenerated back RUBP molecule that was the first acceptor compound for carbon dioxide. So, recalling what happened in the C3 cycle, so the first acceptor molecule for carbon dioxide is the 5 carbon sugar RUBP or ribulose 1,5-bisphosphate. The first stable product that was seen in the reaction is 3 carbon compound phosphoglyceric acid. So, in the C3 cycle, however, this enzyme Rubisco is a carboxylase and an oxygenase enzyme. So, Rubisco enzyme has a dual property. It can behave as a carboxylase enzyme as well as an oxygenase enzyme. So, when it behaves as a carboxylase enzyme, it combines with carbon dioxide when the concentration of carbon dioxide is higher than that of oxygen. But due to the presence of oxygenase activity of Rubisco, whenever oxygen concentration becomes higher than the amount of carbon dioxide in the cell, this Rubisco combines with oxygen, carries out an oxidation process, and that is known as photorespiration. So, in these C3 plants, photorespiration reduces the efficiency of photosynthesis when Rubisco becomes an oxygenase activity. We shall today discuss about how photorespiration occurs and under what conditions. So, in the evolution process, some of the plants have evolved themselves to an alternate path which is called as the C4 cycle or the Hatch and Slack cycle, the second type of fixation of carbon dioxide where the efficiency is much more than the C3 cycle. So, we can see what happens in the Hatch and Slack cycle or the C4 cycle in this slide. So, Hatch and Slack cycle or the C4 cycle. So, this path was worked out by Hatch and Slack, two Australian scientists, in 1967. So, it was found that in some of the plants that are present, the first stable product is a 4 carbon compound instead of 3 carbon compound PGA. So, this 4 carbon compound is oxaloacetic acid. So, the oxaloacetic acid is the first compound that is present. So, this is oxaloacetic acid or the first stable compound of this reaction instead of 3 carbon phosphoglyceric acid. So, phosphoglyceric acid is not the first stable compound in the C4 cycle. So, how has this evolution taken place? C4 plants are generally tropical or subtropical grasses, or their origin is tropical or subtropical in nature. So, they are found in most of the monocots, like you have maize, sugarcane, then sorghum, and some of the dicots like amaranthus and euphorbia. So, what is the condition where the plant has adapted itself to this Hatch and Slack cycle or the C4 cycle? So, basically these plants can photosynthesize at a temperature of 30 to 40 degree Celsius, in high light intensity and are better equipped to withstand water stress. So, these are the basic characteristic feature of the Hatch and Slack cycle. So, Hatch and Slack cycle, how does it operate? So, what are the changes that occur in the plant to carry out the C4 cycle? So, the C4 cycle has a different type of anatomy in the leaf. The C4 plants show a different type of anatomy called as the Kranz anatomy. So, in Kranz anatomy, the C4 leaves are generally thinner. So, the C4 leaves are generally thinner in nature. So, the characteristic is they will have a thinner leaf and with prominent dark vein. You can see the veins are dark and much prominent green in color, and only one type of mesophyll cell and characterized by the presence of tightly packed thick walled bundle sheath cells. So, here take a look at the anatomical structure of the C4 plants. So, in the C4 leaf, so this is the leaf anatomy of the C4 plant. Here this is the upper epidermis, the lower epidermis of the leaf, and then these are the vascular bundles. So, this is the vascular bundle. So, in the Kranz anatomy, the leaf vascular bundles or the veins are much closer to each other and they are surrounded by the tightly packed bundle sheath cells. So, these are the bundle sheath cells that you can see around the xylem and phloem. So, this is a characteristic feature which is known as Kranz. So, Kranz in German means a wreath or a covering or a surrounding. So, they are tightly packed large bundle sheath cells around the vascular bundle. So, these cells are the mesophyll cells. So, these are the mesophyll cells that are present. So, this is the air space beneath the stomata. So, the vascular bundle, this wreath-like configuration is known as Kranz anatomy. So, what is the characteristic feature of separation of finding of these two types of cell? First, the mesophyll cells are loosely packed. There is one type of mesophyll cell instead of palisade and spongy parenchyma. So, you have one type of the palisade or spongy parenchyma cells, and these are large tightly packed vascular bundle cells around the vascular tissue, that is the bundle sheath cell. The mesophyll cell chloroplast, coming to the characteristic feature. So, these are the chloroplasts that are present, and the chloroplast of the bundle sheath cell, the mesophyll cells are granule. So, you have granule chloroplast. The meaning of granule is there is much developed grana inside the cell, and there is absence of Rubisco enzyme in the chloroplast of the mesophyll cell. So, the mesophyll cell chloroplast will not have Rubisco enzyme. So, what we know here is absence of Rubisco in the mesophyll cell chloroplast. That is, the first enzyme Rubisco is absent in the chloroplast of the mesophyll cell. So, instead of Rubisco, they have another carbon dioxide accepting enzyme which is PEP carboxylase, phosphoenol pyruvate carboxylase. So, the first enzyme that will accept carbon dioxide here is PEP carboxylase. What is the characteristic of the bundle sheath cell? The bundle sheath chloroplast will not have any grana. So, they will lack grana, but what they have is enzymes of the C3 cycle. So, in this case you find that there is Rubisco enzyme. So, Rubisco enzyme is present in the chloroplast of the bundle sheath cell. So, here we see there is spatial differentiation, or there is partition of space between the mesophyll cell and the bundle sheath cell to carry out photosynthesis. So, how does this help in the process of dark reaction or the C4 cycle? The carbon dioxide that is diffusing is first moving to the mesophyll cell, then it is taken up by PEP carboxylase enzyme. So, this enzyme, like Rubisco, unlike Rubisco, does not have an oxygenase activity. It is purely a carboxylase enzyme. So, it has been proposed that the presence of PEP carboxylase is an evolutionary characteristic feature to adapt the plant or reduce the loss of photosynthesis by photorespiration process. So, PEP carboxylase is able to concentrate very low concentrations of carbon dioxide, unlike Rubisco, which becomes an oxygenase enzyme. So, let us now go to the characteristic feature or the path of the C4 cycle. So, this is the path of the C4 cycle that you can see, or the C4 pathway. The two green cells here, the first cell denotes the first cell is denoting here the mesophyll cell, and the second cell here is the bundle sheath cell, and this is the vascular tissue or the phloem, in which the product of photosynthesis will be translocated. So, coming to the characteristic of the mesophyll cell. So, initially when carbon dioxide diffuses through the stomata into the substomatal cavity, this carbon dioxide, so the first reaction that will occur in the mesophyll cell. So, the first reaction that occurs is in the mesophyll cell. What happens in the mesophyll cell? The first reaction is carbon dioxide combines with water. So, when it combines with water, it becomes carbonic acid. So, this carbonic acid then dissociates. It becomes H plus and HCO3 minus ions. So, that means here you have fixed carbon dioxide. So, carbon dioxide now is combined with water to become carbonic acid in presence of enzyme carbonic anhydrase. So, carbonic anhydrase is one of the fastest enzyme that is seen. So, after this, the reaction, the next enzyme that comes into action is PEP carboxylase. So, PEP carboxylase now helps in combining carbon dioxide. So, this is the first enzyme. So, the first initial compound that accepts carbon dioxide is now phosphoenol pyruvate. So, phosphoenol pyruvate is a 3-carbon compound. So, phosphoenol pyruvate So this is phosphoenol pyruvate. This is a double bond here. So phosphoenol pyruvate then accepts the carbon dioxide. So it accepts the carbon dioxide. What is formed is, so this is a 3 carbon compound. So 1 carbon is present here. So a total of 4 carbon compound will be formed. This is oxaloacetic acid. So here PEP combines with carbon dioxide in the protonated form. This is oxaloacetate. So oxaloacetate is formed, which is a 4 carbon compound. So what is the importance here? The oxaloacetate compound is the first stable product of the C4 cycle, hence the name is C4 cycle. Then oxaloacetate is then reduced to malate. So here we will transform oxaloacetate into another 4 compound, carbon compound, which is malate. So here so this is 4 carbon compound malate. So after oxaloacetate is formed, so this oxaloacetate is considered as the first stable compound of this C4 cycle. Malate is the next compound that is formed. So here NADPH is utilized. So NADPH is utilized in this particular reaction to convert oxaloacetic acid into the 4 carbon, which is malate. So what happens to malate? This malate is transported through the plasmodesmata. So this opening is the plasmodesmata. So what is a plasmodesmata? So plasmodesmata are intercellular cytoplasmic connections between 2 adjacent plant cells where the cell wall is not present. So this is a continuity between the cytoplasm of the 2 nearby cells. So the mesophyll cells remaining nearer to the bundle sheath cell, the malate is transported through the plasmodesmata into the cell of the bundle sheath cell. So what happens inside the bundle sheath cell? Inside the bundle sheath cell, malate is decarboxylated. You can see carbon dioxide is removed and it gets converted into a 3 carbon compound pyruvate. So we can take out carbon dioxide from here and you will get the next compound, that is pyruvate. So this is the 3 carbon compound pyruvate. So malate, after coming into the bundle sheath cell, gets decarboxylated, releases carbon dioxide, gets converted into 3 carbon pyruvate. And then what happens to this carbon dioxide? This carbon dioxide is accepted by your Calvin cycle enzyme, that is Rubisco. So, rubisco is present in the chloroplast of the bundle sheath cell. So, this carbon dioxide is immediately accepted by rubisco and the cycle of the C3 process or Calvin cycle continues. Glucose or sugar is produced, which is translocated into the phloem for transport. So, why does this type of thing occur inside the C4 plants? So, it is basically an evolutionary adaptation to concentrate molecules of carbon dioxide for rubisco. So, here rubisco will not be coming in contact with much oxygen. So, the carbon dioxide concentrated from the mesophyll cell will be taken up by rubisco. And again in the mesophyll cell, the PEP carboxylase enzyme does not have any oxygenase activity. So, there will be no photorespiration. So, no photorespiration is seen in case of the mesophyll cell. So, here they have prevented the process of photorespiration in the C4 cycle. So, coming to pyruvate, what happens to pyruvate that is formed? The pyruvate that is formed again goes back through the plasmodesmata and then is converted or it regenerates back the PEP molecule, that is the first molecule that was accepting carbon dioxide. So, the change of pyruvate into phosphoenol pyruvate. So, here this pyruvate will require 2 ATP molecules getting converted into 2 ADP molecules and then it will be forming back this compound . So, again the pyruvate, phosphoenol pyruvate will be formed back. So, here you can see the usage of ATP molecule in this step. So, ATP here is converted to ADP. So, initially this pyruvate conversion into PEP is a two-step process where first pyruvate is converted to oxaloacetate and then decarboxylated to form PEP molecule taking phosphate from ADP. So, this process here from pyruvate to PEP requires the presence of 2 ATP molecule. So, for 6 carbon dioxide that can be fixed to get 1 molecule of glucose, the extra number of ATP that is required in this cycle is 12 number. So, this is for 1 carbon dioxide. If we take 6 carbon dioxide molecules, here the requirement of ATP will be extra 12 ATP molecules. So, initially for C3 cycle or for Calvin cycle, the requirement of ATP is already 18 ATP for synthesis of 1 molecule of glucose. The extra number of ATP is 12. So, for this cycle to operate, you require a total of 30 ATP molecules. So, here 30 ATP molecules will be required to carry out the synthesis of 1 molecule of sugar or 1 molecule of glucose. So, what is then the significance of the C4 cycle? So, what we can know of the C4 cycle? So, basically plants having C4 cycle are mainly tropical and subtropical regions are able to survive in environment with low carbon dioxide concentration. So, low carbon dioxide concentration, how they are able to survive? Because PEP carboxylase is going to concentrate very low amounts of carbon dioxide which is needed for the C3 cycle to carry on. C4 plants are partially also adapted to the drought condition, and oxygen will not have any inhibitory effect on the C4 cycle because PEP carboxylase is not sensitive to oxygen molecule. So, because there is absence of photorespiration, the carbon dioxide compensation point is C4, for C4 is lower than that of the C3 plant. So, carbon dioxide compensation point is the point where the amount of carbon dioxide released by respiration is equal to the amount of oxygen that is used up. So, here this is the significance of the C4 cycle. So, recalling how they have maintained the higher efficiency is because these plants are able to carry out photosynthesis at a temperature of 30 to 40 degrees Celsius. So, they are able to carry out photosynthesis even at higher temperature, and they are more adapted to water stress condition. So, these plants basically grow in arid or semi-arid conditions. So, arid or dry or semi-arid conditions also these plants are best suited. So, the tropical grasses like your sugar cane. So, in sugar cane we can see the assimilation of sucrose in larger quantities due to higher efficiency of photosynthesis. You can also find it in maize, sorghum, then you can find them in the dicots like amaranthus and bougainvillea. So, bougainvillea is basically seen planted on the highways due to this property, because it can grow well in the water stress condition and can photosynthesize even under higher intensity of light. So, this is the characteristic feature or significance of the C4 cycle. So, coming to the differences between the C3 and the C4 plants. What are the basic differences between the C3 and the C4 plants that we have learned? So, in the C3 plants, the carbon dioxide fixation will take place only in the mesophyll cell. The mesophyll cell is the only site where carbon dioxide fixation takes place, but in C4 plants we see that carbon dioxide fixation will take place at two places. One is the mesophyll cell and the second is the bundle sheath cell. So, this is spatial partition of reduction process and fixation process of carbon dioxide. So, the fixation process occurs in the mesophyll cell where PEP carboxylase takes up the carbon dioxide. Reduction process into sugar takes place in the bundle sheath cell where there is presence of Rubisco enzyme. So, in the C3 plants, carbon dioxide acceptor is only RUBP. So, we find only RUBP in as the acceptor molecule. In C4 plants, we see the first acceptor molecule is PEP in the mesophyll cell, then RUBP in the bundle sheath cell. What is the first stable product of the C3 cycle. The first stable product of the C3 cycle is 3-phosphoglyceric acid or 3-carbon PGA, whereas the first product in the C4 cycle is 4-carbon oxaloacetic acid. Coming to the anatomical structure of the leaf normally has the palisade parenchyma and the spongy parenchyma without distinct bundle sheath cell around the vascular bundles, and so there is no Kranz anatomy. So, Kranz anatomy is not present in case of the C3 plant, whereas these leaves have evolved to show a Kranz anatomy where there are tightly packed bundle sheath cell surrounding the xylem and phloem or the vascular tissue. The chloroplast character is that the normal chloroplast is present in the C3 cells, whereas in the C4 plant the chloroplasts are of two types, which is dimorphic chloroplast. So, what is dimorphic chloroplast? In the C3 cells, the chloroplast granum is present in the mesophyll cell chloroplast, whereas in the C4 plants, the mesophyll cells have grana. The granum is present in the mesophyll cell and absent in the chloroplast of the bundle sheath cell. So, in the bundle sheath cell, the chloroplasts are agranal, that is absence of grana in the bundle sheath cell chloroplast. So, that is why the chloroplast here becomes dimorphic. So, dimorphic chloroplast is present in case of the C4 plants. The optimum temperature at which the C3 plants best photosynthesize is 20 to 25 degrees Celsius, whereas the C4 plants can photosynthesize at a higher temperatures of about 30 to 45 degrees Celsius. So, why does this happen? When the temperature here increases, the stomata closes. So, in the C3 plants at higher temperature, the stomata closes, and so there is no more diffusion of the carbon dioxide. So, carbon dioxide, when it does not diffuse, the oxygen concentration may increase. So, there may be the presence of photorespiration. So, but in case of the C4 plants, it is more efficient due to no photorespiration. So, the phenomena of photorespiration does not occur in the C4 plants. RUBP carboxylase enzyme is used for fixation, whereas here the enzyme that is used in the C4 plant is PEP carboxylase, and the second enzyme used is RUBP carboxylase. So, the number of ATP molecules that is used to synthesize one molecule of glucose in the C3 cycle is 18 number, and as we saw in the C4 cycle, extra 12 molecules of ATP, that is 18 ATP plus 12 ATP was utilized, that is a total of 30 ATP molecules is consumed. Because there is no loss by photorespiration, this path is considered more efficient. So, it is efficient at low concentrations of carbon dioxide and efficient at higher carbon dioxide. Examples of C3 plants are potato, wheat and paddy, most of the plants, and here you have sugarcane, maize, sorghum, amaranthus and so on. So, these are the basic differences that you have to remember between the C3 plants and the C4 plants. So, next we shall discuss what is photorespiration. See the phenomena of how photorespiration occurs and why photorespiration is also called as a wasteful process in the C3 plants. So, this is what is photorespiration. So, what is photorespiration? Photorespiration is a process which involves oxidation of organic compounds in the plants by oxygen in presence of light. So, photon is light and respiration is breakdown of organic compound, and so here this phenomena is called as photorespiration. So, where is photorespiration found? Photorespiration is found only in the C3 plants and not present in the C4 plants. So, why does it occur? It occurs due to oxygenase activity of Rubisco enzyme. So, it occurs due to oxygenase activity of Rubisco enzyme. So, oxygenase activity of Rubisco enzyme in high concentration of oxygen and low CO2 concentration. So, when the concentration of CO2 is less, the concentration of oxygen is high, then we can see that Rubisco enzyme behaves as an oxygenase enzyme instead of carboxylase enzyme. So, more amount of carbon dioxide is required for Rubisco to function as a carboxylase enzyme. So, the rate of photorespiration is increased when there is bright light and high temperature also. So, in the C3 plants, when the temperature increases, the stomata actually starts to close in order to prevent water evaporation. So, the amount of carbon dioxide that is present inside the mesophyll cell is used up in photosynthesis and oxygen is evolved. So, the oxygen cannot diffuse out of the stomata as it remains closed and the concentration of oxygen increases inside the leaf cells. So, Rubisco there automatically starts functioning as an oxygenase enzyme and then it shows the oxidation process. So, here when Rubisco becomes an oxygenase enzyme, it combines with RUBP, releases one molecule of phosphoglyceric acid, that was a 3-carbon compound. This is a 3-carbon phosphoglyceric acid, and one molecule of 2-carbon phosphoglycolate. So, this is another compound which is a 2-carbon compound, phosphoglycolate. So, sometimes photorespiration is also known as a C2 cycle. So, this phenomena involves 3 organelles. One is chloroplast, the second is peroxisome and the mitochondria. But we say that photorespiration is actually a wasteful process or unwanted process that occurs in the C3 plants. So, what actually happens during photorespiration? So, these are the 3 organelles present. This is chloroplast, peroxisome and the mitochondria. In the chloroplast, this enzyme is your Rubisco enzyme. It takes up oxygen here, then it gets converted into 2-carbon phosphoglycolate and 1 molecule of PGA. This phosphoglycolate is a 2-carbon compound and this 2 carbon compound releases phosphate, inorganic phosphate is released in the chloroplast and it is transported into the peroxisome. So, in the cells of the C3 plants we find that peroxisomes are also prominent in the leaf cells and they are nearer to the chloroplast. So, here phosphoglycolate is converted into glycolate and it moves into the peroxisome. In the peroxisome, glycolate is converted into glyoxylate and glyoxylate gets converted into an amino acid called glycine. So, here hydrogen peroxide is also released in the step where glycolate is converted to glyoxylate. Hydrogen peroxide immediately dissociates and glycine is then transported into the mitochondria. So, in the mitochondria, glycine is decarboxylated, it is deaminated to release carbon dioxide as well as ammonia. So, here it gets converted into another amino acid which is serine. So, serine is then moving out of the mitochondria, it moves back into the peroxisome, gets converted into glycerate. This glycerate then moves into the chloroplast, gets converted into PGA and may enter into the Calvin cycle. So, here you can see there is utilization of ATP in converting glycerate into phosphoglycerate or phosphoglyceric acid. So, even though you are getting PGA in this process, it is consuming ATP molecule. So, it is an unwanted process. So, it is not required by the C3 plants, but initially present as carboxylase and oxygenase enzyme. Rubisco starts showing this phenomenon. So, this is the photorespiration process occurring in the C3 plants. So, because PEP carboxylase does not have any oxygenase activity, unlike Rubisco, there is no photorespiration seen in case of the C4 plants. So, next we shall see what is the characteristic feature or what are the factors that affect the rate of photosynthesis. So, as a physiological process, the physiological processes are affected by a number of factors. So, these factors can be divided into 2 groups. So, one is external factor and also you have internal factors. So, the factors affecting any type of physiological process can be divided into external factors and the internal factors. Let us have a look at the external factors first. The main factor here is light. So, 3 characteristics of light, that is intensity, quality and duration of light, will affect the rate of photosynthesis, because photons of light has to be harvested for the light reactions to occur. So, intensity of light varies. Normally the intensity of light is low when in the evening and in the early morning. So, in the early morning and in the evening, the intensity of light is less and it has maximum intensity at midday. So, here there is a term called as light compensation point. The light compensation point. What is light compensation point? So, it is the light intensity at which the rate of photosynthesis is equal to the rate of respiration. So, when the rate of photosynthesis is equal to the rate of respiration, we say that point is the light compensation point. So, when does it occur? Now, this is reached during the early morning and evening. So, here light compensation point, that is the early mornings and the evening, when the intensity of light reduces, normally the rate of photosynthesis becomes equal to the rate of respiration. So, there will be no release or diffusion of oxygen out into the atmosphere. So, the amount will reduce, and beyond this point, so after early morning, when slowly the intensity of light increases, the carbon dioxide from the atmosphere starts diffusing through the stomata in a much higher quantity, and more photosynthesis occurs. So, there is greater release of oxygen into the atmosphere after the light compensation point. So, slowly towards evening, when the intensity decreases, again the light compensation point is reached. So, coming to the quality of light, the photosynthesis takes place in the wavelengths of the visible spectrum, which is normally between 390 to 760 nanometer. So, maximum absorption, as we know, occurs in the blue and red region of the spectrum, and the normally when the plants in the forest, the upper canopy of the forest generally uses the entire visible spectrum. So, when we come to the lower levels of the forest, the plants will be using specific light wavelength. Similarly, when we come to the organisms that are living at greater depth under the sea, like some of the red algae and the brown algae, they will be absorbing green and mainly blue light. So, these algae start developing more of xanthophyll compounds. So, brown algae, we have fucoxanthin, which is also a compound. Generally, exposure of plants to 10 to 12 hours is sufficient for photosynthesis. So, next factor is carbon dioxide. The atmospheric carbon dioxide concentration is about 0.035%, which is much lower than what is required for optimum photosynthesis. So, in the laboratory conditions or in the artificial condition, when we start increasing the amount of carbon dioxide, the rate of photosynthesis also increases. The yield of many plants like lettuce, which is your salad leaf, the roses and tomatoes, has also been increased artificially by growing them in a carbon dioxide rich greenhouse environment. So, you can increase the photosynthetic efficiency by artificially increasing the carbon dioxide, but very high rate of carbon dioxide or concentration will decrease the photosynthetic efficiency. So, temperature wise, again we find C4 plants can photosynthesize at higher temperature, whereas C3 between 20 to 25 degree Celsius. So, very high temperature will denaturate the enzymes, because the entire photosynthetic process is an enzymatic process. For every 10 degree change, we normally see photosynthetic efficiency also increases up to a basic point, and water is rarely a limiting factor, but in water stress condition, the rate of photosynthesis can also decrease. So, here we come to the end of the chapter of photosynthesis, and with the factors that affect the rate of photosynthesis. Hope you have enjoyed the lecture and understood the process of C3 and C4 cycle of the dark reactions. Thank you all for